Hello friends, welcome to BMH Learning. In this video, we are going to talk about DNA cloning. We all are familiar with the term cloning. When we are talking about DNA cloning, it simply means identical copies of a stretch of DNA. Now DNA cloning is a long procedure, so let's do it step by step. First step is construction of a recombinant DNA molecule. Suppose we have a double-stranded DNA. You can see there is a highlighted portion also. This is the piece or stretch of DNA which we have to clone. So we can call it as DNA of interest. Sometimes it's also called as foreign DNA. Now, the challenge we will encounter is how do we cut this fragment of DNA? So we have molecular scissors which cut the DNA of interest. These scissors are called restriction enzymes. Now, this fragment of DNA is not self-sufficient to make copies of itself because it lacks the required molecular machinery. So what it needs is a molecular vehicle which can deliver it to the host cell for its multiplication. And that molecular vehicle is called as a vector. And the most famous example of vector is plasmid. So what is a plasmid? A small, circular, double-stranded extra-chromosomal DNA. Extra-chromosomal means it's physically separated from chromosomal DNA. The important feature of plasmid is that it can replicate independently of the chromosomal DNA because it has the origin of replication which is absent in DNA of interest. And this is a property which we are going to exploit to increase the copy number of our desired piece of DNA. Apart from this, it has an antibiotic resistance gene. You should note this because this would be very helpful later. Now, in order to put the foreign DNA into the plasmid DNA, first we need to open it. So we bring the same restriction enzyme which was used for cutting the DNA of interest. And then in a process called ligation, which is carried out by the enzyme called ligase, the DNA of interest is pasted, or technically saying it's ligated with the plasmid DNA. Now the molecule we are getting here is called recombinant DNA. Such hybrid molecules are also named as chimera. Now we are getting into the second step, transport into the host cell. So now we have our vector containing the DNA of interest, and it's a time we transfer the vector into the host cell so that the clones or copies of the DNA can be produced. Suppose we have a test tube containing solution which in turn harbors host cells. Generally these are E. coli cells, although other living cells can be used. So we transfer the recombinant DNA into this test tube. Now using some techniques, the bacterial cells are made competent so that they can take up the recombinant DNA. We can show what's actually happening in the test tube is, suppose this is a bacteria, it has its existing chromosomal DNA. And then we introduce the bacteria to our plasmid. Then we use some plasmid transformation technique, and the result is the plasmids are successfully taken up by some of these bacteria. This leads us to the third step, multiplication of recombinant DNA molecule. So within the host cell, the vector multiplies, producing numerous identical copies not only of itself, but also of the gene that it carries. And when the host cell divides, copies of the recombinant DNA molecule are passed to the progeny and further vector replication takes place. So we go to the next step, numerous cell divisions. As we know, in order to increase the copy number of the plasmid, the bacteria needs to divide. So after the transformation, the solution can be poured into a plate containing nutrients for the growth of the bacteria. After a large number of cell divisions, a colony or clone of identical host cells is produced. Some of these colonies have cells which contain copies of recombinant DNA. The gene carried by the recombinant molecule is now said to be cloned. But here we face another obstacle. If you were listening clearly, I mentioned some of the host cells would take up the plasmid, not all. What does that mean? It implies we are getting two types of cells, transformed and non-transformed. The transformed colonies are those in which the cells have taken up the plasmid. But these non-transformed colonies lack any plasmid. It's because they were not competent enough to take up the DNA. You always want that thing which is useful and not a waste one. And for us, the non-transformed colonies are of no use. So the question here is, how do we select them? If you remember, I told you there is an antibiotic resistance gene in the plasmid. Let's manipulate now. So, the nutrient media which we use for the growth of the bacteria, it contains antibiotic. So, the transformed colonies which have the cells that contain plasmids with antibiotic resistance gene, they will survive on this medium. And regarding the non-transformed colonies which lack any antibiotic resistance gene, they will die. So the antibiotic resistance gene acts as a selectable marker. This was all. Thanks for watching.